There's a blood type flowing through the veins of less than 7% of humanity. A genetic anomaly so dangerous it should have vanished from our species tens of thousands of years ago. It kills the children of the women who carry it, defies the basic logic of evolution, and yet hospitals call it liquid gold. Combat medics fight over it. Trauma surgeons reach for it before they know anything else about the patient in front of them. O-negative blood, the forbidden bloodline that nature tried to erase but never could. For most of the 20th century, the existence of O-negative was treated as a curiosity, a footnote in transfusion medicine, useful but unexplained. It did not occur to anyone to ask why a mutation this costly had survived at all. The question was never why O-negative exists. The question is, what was so catastrophic that evolution was willing to sacrifice entire generations of infants just to keep this bloodline alive? Geneticists eventually went looking for the answer. What they found rewrote the history of a species, and it begins with a single deletion, carved into the human genome during the depths of the last ice age, when survival hung in balance. Picture your blood as carrying a kind of identification system. Every red blood cell is studded with molecular markers, antigens, that announce who you are to the immune system patrolling your veins. Type A blood displays A antigens like a badge. Type B carries B markers. Type AB shows both, broadcasting a dual signature. Then there is O-negative, the outlier. Its red blood cells carry none of this. No A antigens, no B antigens, and no Rh factor either. Stripped of every identifying signal, O-negative blood is, in a sense, invisible. It has erased its own molecular identity so completely that almost any immune system on Earth will let it pass without raising an alarm. This is not a small irregularity. The human immune system is one of the most precise detection networks ever produced by evolution. It can tell identical twins apart at the molecular level. It can recognize a single misplaced protein and respond with overwhelming force. It is the reason transplant patients spend the rest of their lives on immunosuppressive drugs, because without them, the body will attack and destroy a transplanted organ as a foreign invader. Against that kind of vigilance, O-negative blood simply does not register. There is nothing for the immune system to find. That is why O-negative is called the universal donor. A person with type A blood, type B, type AB, Rh positive, or Rh negative can all receive O-negative blood without triggering rejection. The mechanism is almost absurdly simple. With no surface markers to recognize, O-negative cells slip past every defensive system the human body has spent millions of years refining. But this universal compatibility comes at a cost that should, by every rule of evolutionary logic, have wiped O-negative out of the human population long before recorded history began. Less than 7% of people on Earth carry it. In parts of Asia, the figure drops below 1%, making it one of the rarest stable genetic traits ever documented. And the deeper question is not how something this rare became this valuable, it is how something this costly managed to survive. On the morning of the 11th of September, 2001, as the towers collapsed, hospitals across New York faced hundreds of casualties at once. Blood types unknown, no time for testing. There was only one answer trauma surgeons reached for: O-negative. Always, only O-negative. That single fact reveals something most people never consider. Every 2 seconds, somewhere in the world, O-negative blood is transfused into a stranger whose blood type nobody has checked. Car accidents at midnight, when blood banks are closed. Mass shootings, where the wounded arrive faster than lab results. Combat zones, where soldiers bleed out before compatibility testing can even begin. In each case, O-negative is the only blood that can be given safely without knowing anything about the recipient. The numbers are extraordinary. O-negative donors make up less than 7% of the population, yet they supply the entirety of the world's emergency blood reserves. Every trauma center, every battlefield hospital, every disaster unit depends on this narrow slice of humanity. Hospitals plan around this fact every single day. And here is the cruelty buried inside that dependence. O-negative donors can give to anyone, but they can only receive from other O-negative donors, trapped inside a supply chain that is permanently, structurally scarce. It is a kind of imbalance built into human biology. History keeps proving how fragile that supply is. During Hurricane Katrina, reserves across the Gulf Coast dropped to a 12-hour supply. The 2010 Haiti earthquake triggered shortages that rippled through blood banks for months. During the 2017 Las Vegas shooting, the city's entire reserve was exhausted within 6 hours, forcing emergency shipments from 3 other states. In Afghanistan and Iraq, O-negative stock determined which forward bases could handle mass casualties and which had to evacuate critical patients elsewhere. Blood banks call it red gold. Military medics call it liquid time. Pediatric surgeons call it miracle blood because a newborn cannot survive a blood type mismatch. But the paradox at the heart of it is this. The same deletion that makes O-negative so valuable also makes it lethal to the children of the women who carry it. In an event many geneticists date to roughly 35,000 years ago, during the depths of the last ice age, though some argue it is far older, an entire gene vanished from the human genome. Not a mutation, but a complete deletion of the RHD gene, the gene responsible for the Rh factor. One generation it was there, the next, in certain populations, it was simply gone. Blood cells emerged that no immune system on Earth had ever encountered before. The timing matters. 35,000 years ago, human beings were small bands of hunter-gatherers, surviving at the edge of what the climate would allow. Every child who died threatened the survival of the entire group. And yet, somewhere in that world, a population was carrying a mutation that, generation after generation, would systematically kill its own children. Here is how the mechanism works. When a woman with O-negative blood becomes pregnant with a baby who is Rh positive, her immune system identifies the fetus's blood as foreign. Antibodies cross the placenta and begin attacking the baby's red blood cells. Doctors call this hemolytic disease of the fetus and newborn. To the people living through it, it must have looked like a curse passed down through certain bloodlines. The first pregnancy is usually safe. The mother's immune system has not yet been primed. But the second pregnancy, the third, the antibodies are ready, and they attack immediately. The fetal blood cells break apart, anemia sets in, brain damage follows. Before modern medicine, the outcome was often fatal. Before a treatment called Rogam was developed in the 1960s, some estimates suggest O-negative mothers lost as much as 50 to 70% of affected pregnancies to this condition, though the precise historical toll is hard to establish. By the cold logic of evolution, this should have been the end of the story. A trait this costly, one that quietly erased a large share of a woman's children generation after generation, should have vanished from the human population. Instead, O-negative spread. It crossed continents. It embedded itself into populations on nearly every inhabited landmass on Earth. Something was protecting it. Something was keeping it alive. Run the numbers, and the problem becomes obvious. If a woman with O-negative blood lost 40% of her children to hemolytic disease, while other women in the same population lost only 10% of theirs to ordinary infant mortality, the math is unforgiving. Within 100 generations, a few thousand years at most, the genetic frequency of O-negative should have collapsed towards zero. Natural selection does not tolerate inefficiency on this scale. A trait that quietly removes 30 extra % of a lineage's offspring, generation after generation, is not supposed to survive into the modern world at all. And yet here it is. Not confined to one isolated valley or one small island population, but present on every inhabited continent, in numbers far too large to be explained by chance, genetic drift, or a handful of lucky founders. Something about the deletion of the RHD gene had to be earning its keep. Something had to be paying for all those lost children. For decades, this contradiction sat unexamined. The reproductive cost of O-negative was well documented in obstetric medicine. The persistence of O-negative across the globe was well documented in population genetics. Almost nobody asked why these two facts could coexist. It was treated as one of those quirks of biology, an oddity to be managed clinically rather than a mystery to be solved. But mysteries like this rarely stay buried once geneticists start asking the right question. If a trait this costly survived, logic dictates that something else, something separate from pregnancy and childbirth, must have been killing people without it. Something brutal enough that carrying the RHD deletion became worth the price paid in the next generation. Whatever that pressure was, it would have to be powerful enough to operate not in one population, but across continents, across thousands of years, consistently enough to leave the same genetic signature behind wherever it acted. In 2011, a team of evolutionary geneticists went looking for that pressure. What they found did not just explain O-negative blood. It reframed the entire history of the diseases that have nearly ended us. Before I show you the last part, if you've ever looked at your own results and wondered where the O-negative bloodline fits into your own ancestry, I put it all together in a guide called Where Do You Really Come From? Link is in the description, but stay right here because what comes next is the part most people completely miss. What they found was not a single disease, but a pattern that repeated itself across an entire family of pathogens. O-negative blood, it turned out, was not an evolutionary mistake. It was evidence of balancing selection operating on a scale that touched the survival of the species itself. Something in the ancestral environment had been killing adults faster than hemolytic disease was killing infants, deadly enough that losing children to incompatibility became an acceptable price to pay. The first clue came from malaria. People with type O blood, the broader blood group that includes both O positive and O negative, show a measurable, statistically significant resistance to Plasmodium falciparum, the most lethal strain of malaria. The mechanism is almost mechanical in its simplicity. Malarial parasites use specific blood group antigens as docking points to invade red blood cells. Type O cells, lacking those antigens, present fewer targets. The parasite has fewer doors to walk through. For the O-negative subgroup, this protection adds to whatever advantage the RHD deletion provides. But malaria was likely only part of the story. Some researchers have proposed that carriers of the RHD deletion may have shown modest survival advantages during outbreaks of bubonic plague, since Yersinia pestis, the bacterium responsible, appears to rely in part on surface markers that other blood types display more prominently. Similar hypotheses have been raised for cholera, where certain virulent strains rely on blood group antigens to invade cells, potentially giving O-negative carriers a narrow advantage. These claims remain less established than the malaria findings. Some have extended this idea to the hemorrhagic fevers. Ebola, Marburg, and Lassa fever are thought to use blood group antigens as part of their cellular invasion strategy, which has led to speculation that O-negative cells might force these viruses onto less efficient infection pathways. Whether this extends to meningitis remains untested. Across at least some of these pathogen families, a recurring pattern has been proposed. O-negative blood may function like a kind of cellular camouflage. While other blood types display molecular flags, some pathogens have evolved to exploit. O-negative cells may move through the body as something closer to genetic ghosts, less readable to infections that target those markers. The trade was brutal, but in evolutionary terms, it was coherent. Lose some children to a quiet, invisible incompatibility, but keep enough adults alive, through plague, through pandemic, through famine-driven epidemics, to keep the species reproducing at all. If the story ended with disease resistance, it would be tidy. But the global distribution of O-negative blood breaks almost every rural population genetics expects it to follow. If a single mutation appeared once and then spread outward through migration, the frequency should fall off gradually with distance from its point of origin. That is not what the map shows. Instead, there are isolated pockets, scattered across the planet, where O-negative frequencies spike to levels that should not exist this far apart. Start with the Basque region of northern Spain. There, O-negative frequencies reach as high as 29%, among the highest concentrations anywhere on Earth. The Basque are not recent arrivals. Genetic analysis confirms they represent one of the oldest continuous populations in Europe, speaking Euskara, a language with no known relatives elsewhere. Their isolation runs so deep that they preserve genetic markers that vanished from the rest of the continent thousands of years ago. Travel south, across the Mediterranean into the Atlas Mountains, and the Berber communities there show O-negative rates of 18 to 25%, despite thousands of years of genetic isolation and centuries of Arab, Roman, and European influence. The Druze populations of Lebanon and Syria show frequencies of 15 to 20%, maintained despite strict religious endogamy, the kind of closed marriage system that should erase rare alleles through simple genetic drift. Move further north to the Celtic populations of Ireland and Scotland, and O-negative rates sit between 13 and 18%, frequencies that predate the arrival of Celtic languages themselves, written into the bones of people who lived there before anyone spoke a Celtic tongue. There is also a deeper puzzle in how O-negative frequencies are distributed worldwide. Indigenous populations of the Americas, separated from Old World genetics for more than 15,000 years, generally show very low O-negative frequencies. The opposite pattern from the Basque, Berber, and Druze populations. That contrast is itself part of the mystery. Either the deletion arose after those populations split off, or it spread unevenly for reasons still not understood. The most disturbing evidence comes not from living populations, but from the dead. In 2022, researchers analyzing DNA from plague-era burial sites in London and Denmark made a striking discovery. Survivors of the Black Death disproportionately carried certain immune gene variants, including one near the gene ERAP2, that alone appears to have increased the odds of surviving the plague by roughly 40%. Across generations, even that advantage compounds. The Black Death killed somewhere between 30 and 50% of Europe's population between 1,347 and 1,351. The survivors were not a random sample of the people who came before them. Certain genetic profiles were consistently overrepresented among those who lived, and researchers continue to investigate whether blood group variation, including the RHD deletion, was part of that picture. Some researchers have proposed that the pattern extends back further still. Archaeological sites from the late Bronze Age collapse, around 1200 to 1150 BCE, a civilizational catastrophe that ended an entire era of Mediterranean history, have been examined for elevated frequencies of Rh-negative markers among the populations that survived it, though this remains a preliminary and largely untested hypothesis. The 6th century Justinian plague may tell a similar story. Constantinople's population is thought to have collapsed from roughly 500,000 people to around 40,000. Whether the genetic composition of those who remained shifted toward O-negative carriers, as some have speculated, has not been established by ancient DNA research. And the trail may go back further still, though this is among the most contested parts of the story. The Toba supervolcanic eruption, roughly 74,000 years ago, has been proposed by some researchers as an event that reduced the human population to as few as a few thousand individuals, though this is disputed. Whether survivors of that period carried the traits that would later define O-negative blood remains speculative. Different centuries, different catastrophes. A pattern that some researchers believe keeps repeating. When human populations collapse under the weight of disease, the survivors who remain may carry a disproportionate share of certain protective genetic variants, the RHD deletion possibly among them. If true, it would be a record, written directly into our DNA, of the near extinctions our species has crawled back from. For 35,000 years, O-negative blood carried a hidden cost. Mothers lost children to an invisible incompatibility passed silently through generations, a curse nobody understood until modern genetics named it. That cost ended in 1968 with a single injection called RhoGAM. For the first time in human history, O-negative carried no penalty at all, only advantages. The trait evolution had been forced to make for 35 millennia was suddenly, quietly, over. And now science is going further. Researchers have begun using gene editing tools like CRISPR to explore engineering universal donor blood cells in the lab. Early stage work that gestures toward replicating, in years, what evolution built over tens of thousands of years through repeated catastrophe. We are no longer just studying this ancient bloodline, we are starting to imagine how to engineer it. If you carry O-negative blood, you are carrying something that has saved lives in every war, every disaster, every operating room in modern history, a genetic inheritance written by every plague your ancestors survived. If you don't, your life may someday depend on someone who does. Today, that is how fragile and how connected survival has always been. Donate blood. The next emergency is closer than anyone thinks. It might be yours. It might be someone you love.